Hello everyone, and thank you for joining today to look at our project. I'm Chris Hovey from NYU Create in the Future Reality Lab. Today, I'm here to tell you about Looking Inside Cells, a suite of virtual reality simulations for learning about cellular biology. Our simulations were designed with 5 guiding principles in mind. 1. Learning is enhanced when students are answering questions through activities. 2. Activities are more meaningful and memorable when students collaboratively construct their answers together. 3. That virtual reality can reveal things which cannot be seen with the naked eye. 4. Simulations are more engaging and hold more learning potential when they represent complex systems. And 5. That designing to enhance common lesson plans will make curriculum integration seamless and easy. Our learning experience designs emphasize meaningful user interactions, collaborative meaning making, and emotionally engaging interactions and audio-visual designs. Looking Inside Cells is broken into 3 main simulations. Build a Cell, in which students use organelles to create their own model of an animal, plant, or prokaryotic cell. Specialized Cells, wherein students modify the generic model of a cell from the first simulation to create real, specialized cells you would find in an organism. Things such as melanocytes, epidermal plant cells, blue-green algae, and more. And lastly, Divide a Cell, where students learn about mitosis by moving, removing, and adding organelles to progress the cellular division. Rather than identifying stages of mitosis in images of cells, students will be able to demonstrate their knowledge by creating images for each stage of the cellular life cycle. Each simulation is inspired by the next-gen science standards and many of the top-rated lesson plans found in their resources. Many of the most popular simulations and labs out there are either two-dimensional or physical models without interactions or feedback. We anticipate that engagement with stereoscopic 3D models will feel more alive to students and improve their mental models of cells. We have designed these simulations to fit each teacher's calendar as easily as possible. The sims can be rearranged or skipped to best fit your lesson plans and use each one as needed. If VR headsets are limited, the class can observe on a traditional screen while students take turns conducting the simulation using a fishbowl-like protocol. Teachers have a few options for debriefing or checking work. Students can use 2D pictures they captured in VR to turn in as checkpoints, give presentations, or conduct peer quizzing with flashcards. Before each unit, you'll want to introduce the topic the way you normally would, with a lecture, reading, or video. Then have students prepare by writing down the things they want to know before completing the simulation. In Unit 1, for example, that will be building a model of a prokaryotic, plant, or animal cell. Once students have completed a simulation, they should reflect on the experience by writing down what they learned, using their pictures from the simulation as evidence. The pictures can then be used in a variety of ways. Students can turn them in for completion and accuracy, use them for presentations, or make flashcards to study with at home. Let's go ahead and look at our simulations. To get started, look at and tap your watch to view the menu and toolbox. Users can interact with each organelle or completed cell in many ways. Once pulled from the palette, an organelle can be moved, rotated, and placed in order to build the target cell. Additionally, users can select an organelle or cell to see its name and a description. In Build a Cell, multiple users and spectators can join the experience. Student teams can do the building, while other students and teachers observe using the PC-based spectator. Our specialized cells include a plant epidermal cell, a blue-green algae, a root hair cell, an E. coli bacteria, a melanocyte, and a red blood cell. In our Divide a Cell lab, we flipped the paradigm of finding cells at various stages of mitosis through a microscope to having students create each image by systematically progressing a cell through the stages of mitosis. In this scene, you can see a user actively advance mitosis by dragging the centrioles to the poles, extending the spindle fibers, breaking apart chromatid pairs, and cleaving the cell into 2 new cells. Finally, we're excited to present our new immunity simulations, currently in exploratory mode with interactive features to come in a future update. The immunity simulations show how protein synthesis is a crucial process during a viral infection. The simulations visualize how T cells and B cells act as part of the immune system to create antibodies and prevent the viruses from entering other cells in the future. While we use COVID-19 as our example virus attacking the cell here, these simulations depict the typical process and can apply broadly to understanding viral immunity. Thank you for watching. We hope you enjoyed this overview of our simulations and look forward to your feedback.